Okay, today I'm going to walk you through how we visualize allelic imbalance in spatial transcriptomics data. Now, as a basic introduction, a diploid cell has two copies of each chromosome, one that was inherited from mom and one that was inherited from dad. These two chromosomes can be distinguished from one another based on the SNPs, or single nucleotide polymorphisms, that make each one unique. So a SNP is a variant that occurs roughly once every 1,000 to 1,500 base pairs, and what make us unique from one another and can distinguish the chromosomes that were inherited from each parent. I'm assuming that you have familiarity with the Integrative Genomics Viewer browser. Shown here is an example of a germline heterozygous SNP. As you can see, these reads are cut off, but roughly half of these reads map to the reference allele, so these would be the entirely gray reads, and the other half map to the alt allele, here being an adenosine. This is also an established 1,000 genome site, so there's quite a bit of evidence suggesting that this is a bona fide germline heterozygous SNP. Now, I'm not going to go into too much detail about how we call these. That's for another talk, but I will mention that if your goal is to visualize allelic imbalance in spatial transcriptomics data, it is of the utmost importance that you first call a high quality set of germline heterozygous SNPs. If, for instance, a homozygous SNP or a sequencing artifact made it into your list, you might erroneously conclude that there was monoallelic expression in the RNA sequencing data, when in fact there was just only one allele to begin with. So this is very important that your heterozygous SNP list be of the highest quality. So as a next step, we want to count the number of reads mapping to each allele from each spot in a spatial transcriptomics array. Here are the sequencing alignments from an example spot. These are not cut off. There are only 4 reads, and you can see that 2 of the reads map to the reference allele and 2 map to the alt allele. So 50% of the reads map to each allele, and you can plot this here on the y-axis as 50% of the reads mapping to what we defined as the major allele, but it's not necessarily important how you define which allele is the major allele. Now, I'm going to recommend that you plot the fraction of reads mapping to each allele as a function of coverage. So here would be 4x coverage, and I think this will become obvious why momentarily. So here is an example of the same SNP, but data from another spot. Ignore the soft clipped read here, and you can see that there are 6 total reads, 5 mapping to the alt allele, which we defined as the major allele, and 1 mapping to the reference allele. Therefore, 83% of the reads here map to the major allele, and the coverage on the x-axis is 6x coverage. So I'm not going to walk through every SNP, every spot combination, but if one does this, your resulting data should look something like this. And I hope it's becoming a little more obvious why we recommend to plot the allelic ratios as a function of sequencing coverage. You'll see that there's this neat pattern for all of the SNP spot combinations shown here on the left. Why is that? Well, this simply reflects all of the various possible allelic ratios that one can get. Let's say you only had 1x coverage. So there's literally just one sequencing read over a SNP site in a spot. Well, that read is either going to map to the major allele, so you have 100% of reads mapping to the major allele, or it will map to the minor allele, so that you have 0% of reads mapping to the major allele, which would, of course, be 100% of reads mapping to the minor allele. And then you can go on and on and on. If you had 2x coverage, you will either have 2 reads mapping to the major allele, 1 to each allele, or 2 reads mapping to the minor allele. So this neat pattern you see here is, as I mentioned, just a reflection of all of the various possible combinations of allelic ratios that one could observe. And it's important to recognize this because I don't think one should be too excited and conclude that there is monoallelic expression when you simply have low sequencing coverage. This would be akin to flipping a quarter 2, 3, 4 times and observing heads every time or tails every time and being excited. So what you really want to see are those SNPs with high sequencing coverage that have an imbalance in reads mapping to each allele. Now, in general, as sequencing coverage increases, the ratio of reads mapping to each allele stabilizes, and this should be expected. This is where the majority of our data points lie. But where's the interesting biology? That would be those reads that are highly expressed, and for whatever reason, they seem to express one allele preferentially over the other. I will briefly mention, you'll have to read our manuscript to learn more about what's going on here, but this was biologically validated. So these are SNPs that map to immunoglobulin genes, and these SNPs were primarily expressed in lymphocytes or immune cells. So it is well established that B cells, which of course harbor an allele from each parent, will undergo rearrangement, silencing one allele and resulting in exclusive expression of the other allele, encoding antibodies. And this is a mechanism so that each B cell only expresses a single antibody. So I will now pass this presentation on to my colleague Li-Ming Chen, who will walk you through the scripts that make this analysis possible.
Okay, in this video, I'm going to show you how to analyze allelic imbalance of spatial transcriptomic data. Before we get started, I assume you have analyzed the DNA-seq data and generated the germline SNPs, and also count the number of alleles of your bulk tumor sample. In our case, we have the output called empire output tumor converted.txt. And we provide an R function called bulk LH plot for you to visualize the allelic imbalance in your bulk tumor sample. For example, you can see here chromosome 3p and 3q. This allelic imbalance matches the copy number variations we observed in the previous video. Keep in mind, what we just talked about is about the allelic imbalance of a tumor bulk sample. Now I'm going to show you how to analyze allelic imbalance of spatial transcriptomic data. The first step is to run a function called LH empire input. This function takes one argument, which is the number of allele of your bulk tumor sample. And it will generate the output called LH empire input.txt in your working directory. This step is optional. And now I'm going to show you how these two files look like. So basically, the function takes this file. This shows you what are the SNPs are. And then this columns shows you how many reads map to each allele. And after running the function, it will generate the output. You will see like this. So basically, it defines the major allele and the minor allele for you. For example, in the column here, you will see the first two SNPs. The alternative allele is the major allele, while the next one, the ref allele, is the major allele. Once you generate this output, now you can run the second step, which is per script. So this script takes two arguments. The first one is the output of the first step. And if you don't run the first step, it will take the germline SNPs list. The second argument is the spot band, which we showed you in the point mutation video how to generate spot bands. And we also provide a customized per script, which you can download here to your local computer. Make sure you need to run this per script for each spot. Once you finish this second step, now we can run the last step. This is a function called LH major allele count. This function takes two arguments. The first one is a list of file paths, which you generated in the second step. The second argument is a numeric number. We showed you how to get this number. This actually is the position of your spot name in your file path. And for example, when you have this file path list, and then you take the first file path and then call this command to get the number. In our example, is 12. When you finish run this function, it will generate two CSV files. The first one is the allele count of all spots. The second one is the filtered version of the first one. And here I can also show you how they look like. Here's the output. You will see which spots have which SNPs and how many reads align to either major or minor allele. With this table, you can further explore the allelic imbalance you are interested. In our example, we generated figure 4 and figure 6. And if you want more details, you can refer to our GitHub page. For example, you will see we are interested in this SNPs, and then we observed the allelic imbalance and generate the plot. I hope this is helpful. Thank you.